Welcome to this afternoon's keynote plenary talk. We're very happy to have Christof Koch here, as he's the president of the Allen Institute for Brain Science in Seattle since 2015. And prior to that, in fact, he was a professor at Caltech, where I did my undergrad degree. In fact, when I graduated, in 1 or 2 years, he came into Caltech. And while I was doing my PhD work at USC, I oftentimes would go back and visit Caltech and have sat in your lecture a few times. So this is a connection. I thought that I want to be the session chair. Anyway, let me just briefly say a few things about him. His undergrad was in physics and philosophy, and PhD. He did his PhD from Max Planck Institute for Biological Cybernetics in 1982, and postdoc fellow at the Artificial Intelligence Lab in the Brain and Cognitive Sciences Department at MIT. Then he became a professor and progressed through. At the time that I think he left for the Allen Institute, he was a professor, and Lois and Victor Chodell, professor of cognitive and behavioral biology at Caltech. I think fairly recently, he has a book on the quest for consciousness, a neurobiological approach, which is very related to the things that we do. His research interests include elucidating the biophysical mechanism underlying neural computations, understanding the mechanism and purpose of visual attention, and uncovering the neural basis for consciousness in the subjective mind. Without further ado, let's welcome Christof.
Thank you very much. Glad to be here at IJCNN, which I haven't done for 2 decades. So I'm not going to talk about consciousness. I'll talk about what we do at the institute I now lead, and the title is Big Science, Team Science, Open Science for Neuroscience. So we all have one of these cool devices in our head. I'm not referring to that. I'm referring to the brain. And what the brain can do is pretty amazing. So if you just try to put some numbers to it, essentially you can compute. They are roughly on the order 10 to the 9 bits. For example, if you do typing on your computer or on your phone, that we all do obsessively for many hours each day, and seem to derive pleasure from that. We have 10 to the 9 bits per second that stream in, roughly at this sort of light level, through our cones. And we can, people have done estimate order of magnitude in terms of typing. Typing speed, speaking is roughly the same. Given the entropy, it's roughly 10 bits per second of motor information. So in other words, we can take in 10 to the 9 bits of input and translate that in real time into 10 bits of output information. It's highly trained, lots of experiments shown that, because we do it every day for so many hours. And the same visual system, of course, can be rapidly deployed to go talk, to eat with a fork, or to recognize somebody famous. And so the challenge is, all of that is done with 10 to the 11 neurons that fire roughly 1 trillion spikes per second. It's a good number to remember, 10 to the 12th action potential per second. Of course, many of those are in the cerebellum, not in cortex, but still, on the order of 10 to the 11 neurons, 10 to the 14 synapses, generating 10 to the 12 spikes. And so that is sort of at the biophysical level that we study. That's sort of the underlying machinery that seems to translate all the visual input into output information. We study 2 brains. You can see them there. That's a human brain, and then almost exactly 1,000 times smaller, it's the brain of the mouse. It's 1,000 times smaller in terms of volume, area of cortex, or number of neurons. And what cortex is different, so there are a number of different types of neural tissue. Cortex is unique in fact, it's the defining characteristics of mammals. So one of the features that defines mammals is the fact that they have a cortex, particularly a neocortex, something evolutionary new that came out on this stage 210 million years ago. It's essentially 2 plus epsilon dimensional technology. So in our case, human brain, it's the thickness of my jacket, 2 to 3 millimeters. It's roughly this size. It's 1,200 square centimeters, so it's like a pizza, medium-sized pizza. You've got 2 of those sheets, and they're rolled up. If you're an adult, you've got 2 of them, and they're rolled up, and they make up your part of your forebrain. That's cortex. It's 2 plus epsilon dimensional because the width here has changed only by factor 2 to 3. Let's say between a mouse where it's 800 micrometer to us where it's 2 to 3 millimeters. If you look across the entire mammalian kingdom, it's changed by factor 2 to 3 versus the area has changed roughly 100,000. So that's what I mean. There's something constant about this tissue. It's really the thing that seems to vary in different animals, including us, is the total areal extent of this tissue. So the question we and many other people are pursuing, there seems to be something particular powerful in terms of computational capabilities in a little bit of canonical tissue. And so the question is there, across the different bits and pieces of the brain, the human brain and the mouse brain, is there some canonical operation that we can understand, decipher, A, to help people if they have diseases of cortex, or B, to build better machines. Now the remarkable thing is, so here you have a so-called Nissl stain, a stain that was developed at the end of the 19th century that shows up structures in the cell bodies, whether they're neurons or supporting cells. One of these is the mouse brain cortex, the other one is the human cortex. They look very similar. Once I remove the scale bar, in fact, they become almost impossible to distinguish. Now, so roughly, if I go back here, one of the other numbers, interesting number to remember, typical, if you take a class from me, is something I usually ask, how many neurons are there under cubic, how many neurons are there, how many neurons are there under a square millimeter of cortex? There are roughly 100,000. There's something, this thing is going in and out. There are roughly 100,000 cells per under a cubic millimeter. So depending on the thickness, there could be anywhere between 50 and 200,000 neurons in that column. Now it turns out, we first discovered this in the early 19th century. People discovered that all biological organisms consisted of cells. Cells is sort of the basic unit of any biological organism, whether it's a single cell organism or an organism like us with 40 trillion cells. And cells, of course, beget other cells using, and they do their DNA and RNA and proteins and all of that. Now, we know also since the 19th century there are lots of different types of cells. There are liver cells, and there are cornea cells, and there are brain cells. They're very different, although they, of course, they all carry the same genome. They express different genes, obviously. Now, even if you look at brain cells, it's not just a homogeneous soup like a Wij matrix would suggest, or Hopfield network, or general connectionist approach would suggest. There are many different types of neurons. They're not just 2 classes of neurons, excitatory and inhibitory neurons, but now we estimate they're probably on the order of 1,000 different cell types. 1,000 different cell types. Each cell types differ in their function, in their electrical behavior, in the gene they're expressed, in where they're situated, who they get input from, and who do they talk to. And understanding brains, it's going to be essential to try to understand how many cell types are there, how do they relate to each other, and how do they instantiate different computation. If computation is the best metaphor to think about the brain, which is a question that remains open to this day. So these are just some of the cell types that people have discovered. Again, this goes back to 1880, 1890, Golgi and Raymond y Cajal, sort of the patron saint of biology, who first drew all these different neurons. There's some very distinct shapes. Now, once again here, in fact, I showed this test to my institute, 300 people, where I showed 12 neurons, each neuron like this, and I asked them to guess which one is human and which one is mouse. Once you remove the scale bar, so as I said, the human cortex is 3 times thicker, so the neurons are 3 times longer, roughly. Other than that, the neurons are very similar. They're not identical. There are lots of specialization, but there isn't anything magic different about the human brain from the mouse brain. In fact, if you look at across different mammals, dogs, cats, mice, rats, whales, only an expert, really, armed with a microscope and precise knowledge can tell whether this comes from a dog or mouse or monkey. The basic stuff is all the same. We just have less or more of it. Now we of course don't have the most. The most has animals like the whales or like dolphins. They have cortex that's significantly bigger, including having twice as many neurons. A little bit embarrassing for us because we think we're always the best, of course. In fact, here I have that. So here's some interesting numbers. They said roughly factor 1,000 between human and mouse. The pilot whale actually has an embarrassing 37 billion neurons compared to the 16 billion neurons, cortical neurons we have. We have 86 billion in our entire brain, but almost 80% of those neurons are in the little brain at the back, the cerebellum. Right, so what's the challenge? The challenge over the last 100 years of neuroscience has been that we have systems that are very, very different from the systems that typically physics studies. So what physics has done best, at the extreme small or the extreme large, quantum mechanics or cosmology, physics study systems that are highly homogeneous, that have very simple boundary conditions like infinite or far away or stationary distributions that have large number of very homogeneous particles. The brain couldn't be more different. So the brain has large numbers, depending what brain you're looking at, you're talking to 10 to the 7 or 10 to the 11 neurons or 10 to the 12 neurons. They're highly heterogeneous, as I said. They're probably easily 1,000 different cell types, interconnecting with very large numbers of other neurons. It now turns out that now we have more detailed information about connectivity available. The connectivity, unlike, for example, a Facebook graph, is quite dense. So you might have neurons that connect to 50,000, 100,000 other neurons. And their weight isn't just all or none, but sort of looks more like it's log normal distributed. So it makes it very heterogeneous to study. And of course they interact, again, unlike in physics, across a multitude of scales, right? From the level of millisecond or sub-millisecond synaptic release to, of course, years in our case, from sub-micrometer, again, from the level of synapses all the way to tens or, a meter if you're going from here down the spinal cord. We don't have a complete list of the cellular components. That's one problem. We don't know how many cell types there are. We don't know how they relate to each other. It's very difficult to record for more than a very small fraction simultaneously. So typically, neuroscience has done one of two things. Either neuroscience looks at bulk tissue, right? So you go into a magnet, and then you do hemodynamics, which is really cool because you can do it in normal subjects. It's very safe. But what you get is a blood flow. So you get the power, essentially the blood flow to a large area of the brain. A small voxel in fMRI includes several million neurons of 1,000 different or 100 different types maybe. Or neuroscience is very good at recording from small groups of individual cells using electrode or 2-photon calcium imaging. But so imagine you're watching a movie. Imagine you're watching the latest Hollywood movie and all you can see is 0.01% or 10 to the minus 6 of all the pixels. Right? So imagine you're going to the cinema, in lieu of having the entire screen, you have a pixel here, a pixel there, and a pixel there. You're now trying to understand what goes on, the underlying action of the movie, and who loves whom and who backstabs whom based on this individual pixel. This is sort of the situation we are facing in neuroscience. The very few accepted standards in neuroscience, every lab has different standards, even for such obvious things called as action potential or 40 Hz oscillation or spindles or sharp waves. Unlike other sciences, there's no central unifying project, although that's very slowly changing. And in terms of the academic landscape, they're on the order of 10,000 labs. There are roughly 60 to 70,000 neuroscientists worldwide. And they live in the sort of their ecosystem are 10,000 labs. I'm sorry. All right, is this better? Okay. So you have on the order of 10,000 different labs, and oh, hi, Chava. I was looking for you. Okay, I think I should just stay away from here. They're on the order of 10,000 different labs, and if you've ever been to Neuroscience, Society of Neuroscience, 35,000 people, you have this feeling, it's a sociological Big Bang, where everybody's moving away from everybody else at the speed of light. Why? Well, because they're using different animals, different tools. Some are using old, some adults, some very young, different parts of the brain using radical different techniques, some disease, some healthy, and so it's a very challenging situation. It's very exciting, but on the other hand, neuroscience is now leaving its romantic phase. So the first 100 years, a romantic phase where you're just beginning to discover the lay of the lands, like the early exploration of, for example, this country here in Alaska. But now it's entering a more mature phase where there are a large number of people, they get a large number of money. I mean, neuroscience alone in the US is supported to the tune of $5 to $6 billion a year. And so the taxpayer who funds most of it, not all, but who funds most of it, wants at some point results, like, can you help my grandmother who's beginning to suffer from Alzheimer? Or can you help my kid with ADHD? Or what about somebody who is depressed? Or what about somebody who hears voices? Can we do something about them? And the sad fact of the matter is right now we can't. We're unable to do much of that because we're still at this very early phase. And finally, universities are fantastic. They're one of the best institutions the West has discovered or has perfected over the last 900 years. But they're not set up for large-scale systematic efforts. That's something that universities simply don't do, which is why, for example, there are large physical observatories like telescopes or colliders that are built independent of, like at LHC in Geneva, that are built independent of university. So we're trying to occupy a little bit this space here. We're not trying to compete at all with universities. We're trying to do something that's quite different that can't be done at the university. So what are we? So the Allen Institute for Brain Science is an independent, non-for-profit. Everything we do is non-profit. A medical research organization started roughly 14 years ago, and our goal is to accelerate neuroscience. It's as simple as that, accelerate, fuel neuroscience. We roughly right now have 300 employees. We're growing to 400. And our culture is somewhere between the universities, so people like me and many employees come from universities, motivated by basic science. You know, we're trying to understand the most complex, organized, piece of highly active matter in the universe. But then we have a lot of DNA coming from biotech. Some of our people come from biotech, and we have things like SMART goals. Most of you probably don't know what that is, which is a good thing. SMART goals and program managers and tight deadlines and things of that sort that you don't have at a university. And we also don't have tenure. So it means we have much more flexibility to recruit people or to let them go if things don't work out. When I came from Caltech in 2011, together with Alan Jones, we went to Paul Allen, our donor, and proposed a 10-year plan for roughly $1 billion for up to 500 people that we're executing now. So this plan goes from 2012 to 2022. So we are 5 years into this plan. To build these observatories, I'll tell you about for mice and for human cortex. At this point, we don't do any other species. That's roughly our budget, and this is our donor. Paul Allen, remarkable man. As I said, everything we're doing, this is our 10-year plan. He also gave us a gorgeous new building just next to Amazon, if you know where that is, in the South Lake Union neighborhood of Seattle, beautiful new glass building that he gave to us. So we are about big science, team science, and open science. So what I mean by that? Big science, I mean, we try not to compete with universities. There are wonderful universities out there. There's no need to have yet another university department. So we do things that can only be done at large scale. We do them in tightly organized teams. This is our biggest challenge. Really how to organize teams where we all work together using very similar tools so things are comparable, and there's synergy. We try very hard to be more than the sum of a bunch of individual people. And finally, open science. So from the get-go, all of our data is accessible. All of our data, our SOPs, our transgenic animals, our tools, all of that is available. You can right now go to our website, brain-map.org, and you can download everything I'm going to talk about. And if you're from Big Pharma and you make some discovery, wonderful, and we hope you'll acknowledge us in your paper. All right. So why do we study the mouse? The mouse, actually here. I don't know if you can see it. This is actually a model of the mouse. It's accurate, anatomically very accurate, 3D printed. This is actually 8 times larger volumes, twice as large linearly. So the mouse brain is smaller than a sugar cube. One of its great features is it has a smooth cortex. So everything is accessible, right? Compared to a human that's highly convolved, of course. We have fantastic technologies, you will have heard some of them, things like optogenetics that allows us to intervene wherever we want, pretty much wherever we want in a given population, as long as we know its genetic zip code, as it were, we can address it and we can turn things on or off with millisecond precision. That's just a fantastic technology that's really not available in monkeys and not at all for safety humans at this point in time. So that's probably the biggest reason. We are very good at building, we both at the institute as well as the field as a whole is very good at manipulating the genome of the mouse, so we can do all sorts of things. We can get individual neurons to glow, so you can see them. We can we can turn, again, using all this technology, we can very deliberately interfere at a particular point in time in the development of the animal, or a particular point of your choosing when you want to turn off neurons now. That's really, there's no other mammal. That's possible in a fly, but not in a mammal, except the mouse. It's small enough so we can model it completely, yet it's structurally similar to the human brain. As I said, we all need as children, and a piece of, if I give you a little quinoa-sized piece of human brain and mouse brain, nobody here could tell which one is which. It's very similar. All right, so that's the reason why we study the mouse brain. So our big goal is we want to understand, we're putting a lot of resources into understanding this quinoa-sized grain, 1 by 1 by 1 millimeter, of an adult mouse. So it's a standard lab mouse. They're now used very, very widely, called C57 Black 6J. We make all of this available. As I said, we focus on the visual system. We have to focus on some system, given my background and the fact that we have very powerful models of vision in other mammals. We choose to focus on the visual system, visual cortex. So there we are trying to understand how it works. This, as I like to say, the most organized piece of excitable matter, using a bunch of different focus. All these different projects are sort of implemented in terms of pipelines that generate large data flows using lab information management systems that are highly tightly synchronized and that have underlying